Hi, it's Leslie Farouk at American School Foundation of Monterey, taking a little time to recap on the AP biologists' activities in the past week. 2 weeks ago, I exposed them to a guided version of the AP Biology Inquiry Lab number 5 about photosynthesis. And after we determined it was possible to squeeze the spongy mesophyll such that the leaf discs would sink, I asked them to generate some ideas as to what variable they could test for. They came up with a variety of different variables, as you can see here, and on the double block of the past week, I told them to bring everything they needed and that the time would be theirs to conduct their open inquiry experiment. One group opted to vary the concentration of the sodium bicarbonate in distilled water, and definitely it came in handy that they are concurrently enrolled in AP Chemistry. They were able to calculate how much solute they needed to dissolve in order to obtain these molarities, and in addition, some of the student groups had a very methodical setup, almost as though it was an assembly line. Of course, the trickiest part of this lab would be getting that level of pressure and suction just right on the syringes in order to get the leaf discs to sink, but to not damage the leaf tissue itself. When I checked on the students in their sunspot in the hallway, I could see that they were making great use of their cell phone timer app, and they taught me that it's possible to hit the lap button every time a leaf disc floats, and then you have very accurate data as to exactly how many leaf discs over time were floating in the various treatments. And of course, as is the case with science, some groups had greater success than others with their given variables, and some groups, like the group that decided to vary the light color reaching the leaf discs, have to do a redo, possibly with a thinner layer of cellophane because they couldn't generate any floating discs at all. So they're going to be coming in for that this coming week. This was what we were hoping to achieve when we started out. And fortunately, 4 out of 5 groups were able to get rate of photosynthesis data based on the flotation of these discs the first time around. I was very happy that I decided to undertake this particular AP biology lab. Lots of great learning and lots of hands-on inquiry during this particular lab activity. Thank you for tuning in.